So yes, thank you, Robbie, and to your group for this workshop. So as you said, we work with Eugene and your group, the group in Liverpool, on this project about aging and intercellular communication. So we built an atlas of changes in intercellular communication with age called scAgeCom. So let me start. So the main objective at the beginning was really how can we quantify changes in intercellular communication with age? And more broadly, can we then interpret the results to maybe learn about gene regulation and tissue homeostasis? Because intercellular communication is just the interplay between this and in the middle, if you want, in terms of biological level. When working on this, we started by looking at single cell data, and we use single cell data to infer those changes. And while doing so, we thought we might create a package that would work not only for aging, but to compare changes in intercellular communication between any two biological conditions. So we created scDiffCom. That's our package that I will present. And then we applied this package to aging data, mouse data, to build an atlas of intercellular communication changes. So I will go with scAgeCom and present a few things, and then Eugene will go with the rest of the talk. So the first question is I want to answer is how can we try to measure intercellular communication, and there more specifically changes in intercellular communication? Because when I started postdoc, there was more tools were developed to work with single cell data, including tools to try to detect communication patterns between cell types, but not to infer changes between these patterns. So, and at the same time, there was also a lot of more and more aging data. So we thought we could combine them together. So the way it works, the package works, is at first we use single cell transcriptomic data. So on this t-SNP plot, for example, each dot is a cell. And what we know, the type of data we have, that for each cell, let's say we have n cell, we have the expression of all the genes we have measured, right? In some normalized counts here. And with that, you can do a lot of things. So you can first classify your cells according to cell type, which is something that needs to be done before we applied our package. So here, for example, we see that there are 4 cell types based on the transcriptomic profile of these cells. And we also need to have 2 conditions, which in our case would be young and old, but it's more generic than that here. So let's imagine you have young cell and old cells, and we want to try we want to do some differential expression, not only gene by gene, but by communication. So what we do first, we average the gene expression per cell type and per condition. So let's say gene 1 has an average expression of 1.2 in the young cells of cell type 1, B cell or whatever. What we do then with that is to infer communication patterns, we need to have some information about how cell communicates. And we use database of ligand receptor interactions. So I will come back to that later, but we have this database now of around 5,000 interactions. Let's say, for example, ApoE is known to interact with LDLR. So the idea is now to try to see how ApoE is expressed in these cells and how LDLR is expressed in these cells, and try to infer if there might be some signals between particular cell types. So what we do is that we kind of first build all interaction we can imagine. Let's say you have 4 cell types. So you can imagine 16 interactions, cell type 1 to 2, 1 to 3, 4, etc., etc. And for each of these 16 cell type interactions, you can look at the expression of all the ligand receptor pairs you have, so 5,000 of them. So you get like a large list of hypothetic interactions. And you assess, you start to compute scores for them. So score of condition 1, condition 2, so young and old, and you try to see if this change. But now the important part is to find, okay, are these signals really biological? And then do they really change, or is it just noise? So we perform a series of statistical tests, permutation tests, to first, okay, see if the score in young is very significant or not. Same thing for old or condition 2 if you want, and then the difference. We combine these kind of things together, and based on all these values, the p-values we get, the score, the log-fold change, we end up with the algorithm at the end to classify each interaction, which is made of these two cells, so cell type 3, cell type 1, for example, and a pair of ligand receptor. We are able to say, okay, if this is staying stable, if it's upregulated, downregulated, or simply not detected with A. So this is the basic idea of scDiffCom and what it does. Now we have a few, I want to give you one slide about how we built the ligand receptor database, okay? So this was quite important. We used previous knowledge from 7 other papers that curated such interactions, and that also built tools, by the way, not to detect changes, but more, they all have their own specificities, I would say, mostly to try to detect interactions. So here you can see the name of this one, like self-DB is one which is quite known, quite popular, and they all have their own, if you want, list of ligand receptor pairs. Sometimes it's more than a pair, sometimes it comes with heteromeric proteins, so you have to combine 2 genes together to make this more biologically accurate. So when we combine all that, we obtained around 5,000 mouse interaction at the end, roughly the same for human. We still have to remove a few of them that we think were miscurated based on some papers that were, what people thought it was intercellular communication, but it was mostly intracellular signals. But that was like marginal corrections. So you can see here like how they distribute according to the 7 database. Okay. Another thing we did, which was important before we could really start interpreting results, was try to annotate these interactions with Go terms. Because okay, we have Go terms attached to each gene, but what we wanted to do is really attach Go term and cake pathway to the pairs themselves. So on this graph, what you can see is that, let's say you have ApoE LDLR and you have all the Go terms for ApoE, you get a graph which will be the blue graph of the ligand graph. Okay, so these 3 nodes, these 4 nodes here will be the Go terms, and then you have all the parent terms. You do the same thing for the receptor, and then what you get is you take the intersection in common in between the graph. And you do that because usually genes are annotated with the lowest, I mean the lowest level or highest level, if you want, terms. So if you were just doing the intersection, you would just get nothing here, just this term, right? But if you take the parents, you really see the process which are common in between the 2. And that's the way we got around that. So we have then a list of Go terms attached to each of our pairs. And the last was reason why we did that if we remember that we have this list of detected interactions, what we wanted to be able to do after that, because we kind of got a lot of them, as you will see, was try to, okay, can we extract dominant signals in that? So in the tool itself, we came up with some overrepresentation test. So let's say on this plot, that you have a volcano plot. So just, this is not a usual gene volcano plot because each point is not a gene. Each point is made of 4 components, 2 cell types and 2 genes, right? And the idea is, okay, let's say that all these interactions in red are upregulated. Do we have some gene of some ligand receptor which is more overrepresented, like in the red points than the other points? Meaning that it will usually take part into interaction between cell types and where they are upregulated with age or whatever condition you are comparing to. So we do a Fisher exact test for that. And we are able at the end to return, like for example, ApoE LRP1 here, we see that it's 10 times up and 13 times not up, but usually you have a much more, you don't have a lot of upregulated signal in this tissue. So it means that usually this ligand receptor is usually more read by chance than anything else. So you get some sort of ratio and p-value. Then you can do that with everything. Genes, cell types, like for example, are B cells mostly located in the red panel than the other one? And you can do that and say, okay, maybe B cells are usually involved in process upregulated with age. And we did that with Go terms as well, etc. So this is the package itself. And now I want to move to scAgeCom, which is where we apply that to aging data. So I will present the first slide, and then Eugene will go and present the details of what we got. So when I also started the product, that was just a few weeks after that, there was a bioarchive paper called Tabula muricenis that appeared, which has now been published. When they released like 23, 25 tissues with single cell data, aging data for them. So then we use all of these tissues that you can see here, adipose tissues, etc. And we tried to apply our tool on it. Actually, it was more the opposite. We applied our method on that, and then we created the tool because we thought we could do both. But we generated like data for 58 data sets with another data set from Calico for comparison. So now I will let Eugene proceed to, okay, what we got from there. So I don't know if you want to present your slides or if you want me to just keep going with that. So let me know.
I think it would be better if I would share just to.
Yeah, go. Okay.
Thanks. Thank you, Cyril. Thanks to everyone for the workshop and for the opportunity to present our work here. So our app scAgeCom presents two categories of results. On one hand, we have results that are specific to a particular tissue, let's say liver, and we call that tissue-specific results. And we also have results that are derived from multiple tissues, which we call cross-tissues results or global results. So for the tissue-specific results, the app presents multiple ways of getting insights. For example, Cyril already talked about the volcano plot, which is presented in panel A and which displays all the cell-cell interactions within a tissue. Panel B presents a different kind of plot, which shows all the cell-cell interactions from a tissue from the perspective of the cell-cell interaction score in the young and old sample groups, from which one can detect the differences from another perspective. And the panel C shows another representations in terms of the receptor and ligand log-fold change between the two conditions, from which one can get an insight into whether the receptor, the ligand, or both drive the aging modification of a particular cell-cell interactions, both for particular ligand, for single cell-cell interactions, but also for patterns on a whole tissue scale. Other kinds of tissue-specific results come from further downstream processing. So, for example, panel D presents a type of network representation of the cell type pairs, from which one can derive the major cell type pairs that drive the signals within a tissue. Panel E shows a tree map representation of the gene ontology terms that are most importantly observed in the results for a tissue. And panel F shows the top patterns, so the sorted top patterns in a particular tissue, where pattern could mean either a ligand receptor interaction or a gene ontology term or a KEGG pathway. For the cross-tissue results, these are presented both in a tabular format, as is presented on the current slide. So, for example, one could check upregulated geo terms such as immune response, and from the table, the user can see that this particular pattern has been observed in 19 out of the 23 tissues. And further insight can be gained by examining the same kind of counts for categories of data sets obtained by different experimental single cell techniques. If one wants to get further insight into a particular pattern, such as upregulation of T-cell differentiation, one could get so by examining which results from which data set drive the particular pattern. So in this particular representation, one could see that the overrepresentation of T-cell differentiation in all of the data sets that have been analyzed. And the color code here is showing that there is a lot of red rectangles, which indicates that the T-cell differentiation has been detected as upregulated in many data sets coming from various tissues and various single-cell techniques, and also sexes, sample sexes. The app itself is developed as a Shiny app served through Shiny proxy and is available at scAgeCom.org. So if you're interested, we invite you to check it and use it. So I'd like to also provide a short overview of some of the selected results and their interpretations from the aforementioned analysis. An important category of result is basically upregulation of immune response in general and beta-2 microglobulin or B2M in particular. So the representation described before for T-cell differentiation indicates that it is detected as being upregulated in multiple tissues across multiple dataset categories. And this result is mainly driven on further analysis by B2M interactions, namely B2M interactions with CD3G and CD3D, which biologically correspond to an interaction between major histocompatibility complex of type 1 with T-cell receptor, which suggests that potentially with age, antigen presentation via MHC1 may increase. B2M in general has been described in the literature as being associated with aging, both on the gene expression level as well as in terms of levels in circulation. An important note is that such studies can describe results that drive aging on one hand, but also can indicate changes that come from compensatory mechanisms. And two relevant examples in our results are the interaction MIF CD74 and HMGB1 thrombomodulin. In particular, MIF CD74 is an interaction that has been described to have the possibility to rejuvenate MSCs from particular kinds of induced senescence, while HMGB1 in general is considered to be a pro-inflammatory factor, while its interaction with thrombomodulin is considered an anti-inflammatory mechanism. So both of these are detected as upregulated in our analysis and might indicate activation of compensatory mechanisms with age. Another important category of results is represented by the downregulation of many molecules such as integrins and collagen across many tissues and many data sets, as indicated by the blue boxes. And this has also been a widely described result in the aging research. To provide an example of a slightly more in-depth analysis at the tissue level, we'll give an example on angiogenesis, and in particular on the interaction of GPI1 and AMFR in the pancreas. So the table presents the GPI1 and AMFR interaction, cell-cell interactions directed toward the endothelial cells from other kinds of emitter cells, such as pancreatic cells and leukocytes. And all of them are shown to be downregulated with age. The plot can show that this pattern comes mostly from the downregulation of the receptor expression, as shown by almost fourfold decrease. And the network representations can provide further insight in showing that in general, communication towards endothelial cells from multiple cell types is downregulated, with an important exception of leukocyte to endothelial cell communication, which is, roughly speaking, upregulated, but which makes sense in the context of the previous discussion about the upregulation of immune response and the general known fact of phenomenon of inflammation. Another important category of results relates to lipid metabolism. Namely, we detect upregulation of ApoE, which is not presented in this given slide, but we chose to present another important finding in this slide, namely the global downregulation of APP, amyloid precursor protein, observed at least in TMS-FACS male category of datasets. This was interesting to us because we've been able to observe that APP, which is a molecule generally discussed as being involved in Alzheimer and brain pathology, degenerative brain pathology, is actually expressed in many tissues and is actually an important signal that we detect to be downregulated with age. And there are reports and research results that indicate association of APP with pathology in tissues other than brain. So to conclude, we would like to say that scDiffCom offers the first statistical method to properly infer changes in intercellular communication between two conditions and by extension between multiple conditions. The scAgeCom application is available for the community. It contains numerous results waiting to be interpreted. And not only our results support previous knowledge regarding aging, they provide a new level of details and have the possibility of generating new hypotheses to be tested in the lab. Last but not least, I would like to acknowledge the great team I had the opportunity to be part of and to work, including people from University of Liverpool, Institute of Biochemistry, Karolinska Institute, and Jan Zuckerberg Biohub. Thank you for having the opportunity to talk here. It was a great pleasure, and we'd like to clarify any unclarities that may have arisen during the call. And we do invite you to check our app available at scagecom.org. Thank you.
Thank you, Cyril, and thank you, Eugen. This was a great talk. And I believe we also have a paper already pre-published, right? I don't know if you mentioned this. So people can check the information about the app and the results in more detail there. If there is maybe one or let's say not two, one question quickly, we can have time for it. If not, we could move on. So any questions for Cyril or Eugen?
I was going to make a quick question, but I think the answer might be long. No,
we are flexible. You know what? Go ahead.
No, I'll just put it out there, but we can discuss in more detail later. But it's actually more about APP and what you were showing about its expression in different tissues. I mean, there's been some others pointing that out. You know, why does it cause more pathology in the brain? And it's not well understood. But you said that it's been shown to cause pathology in some other tissues besides the brain. Do you know which ones?
Yeah, so we had a few slides on them. We didn't put them for time limitation, but I mean, if you don't mind, Eugen, I have a few in mind. So that's so there are papers relating APP and obesity. So they look at obese people and they look some abnormal expression of APP in adipose tissues. There was also a link between APP and in the intestine, and expression was modulated by diet, which I found interesting because diet is quite important in aging. There was also a role of APP at neuromuscular junctions in limb muscles. And there was another paper on skin, but I don't remember which pathology exactly. But actually, there is one paper that says, like, okay, APP is not well studied outside the brain, and it's listing these few papers I just talked about. So we looked at them, and there are actually 5 or 6, but I think there are not so many other studies that looked at them. But there are quite a few, like I just said.